In this video, we'll continue our investigation of how, using an evolutionary perspective, we can learn more about illnesses that affect humans. Let's first begin by investigating the evolution of the influenza virus. Normal flu season, we see the death of about 20,000 Americans. But I'm sure you've all heard of the Spanish flu of 1918. This is a huge pandemic, killing 50 to 100 million people, which affected about that time 20% of the world's population. So there's been coevolution between humans and influenza viruses, and hosts can fight off an infection if they're able to recognize the antigenic proteins on a virus exterior. Now that recognition comes from a previous infection. So, in the coevolution between humans and the virus, any virus whose antigenic proteins evolve more quickly are more likely to be able to hide from a human's immune system if they've been previously exposed to an influenza virus. So we would predict that a successful virus would evolve more quickly at these antigenic sites. And sure enough, if you look at lineages in a next year's flu season, those that survived into that next year, 76.7% of these viruses had mutations in their antigenic sites versus those lineages that were in the last year that are not successful really establishing an outbreak during the flu season. They only had 47% of those had mutations in the antigenic sites. So sure enough, more successful invading viruses are those that are evolving more quickly at these antigenic sites, so they can more successfully hide from the human immune system. And we know that there are 18 codons specifically that when they change, it tends to be more successful for the virus. So what this allows us to do is to look at those viruses that have established those changes at the end of one previous flu season and develop vaccines based on those viral codes. So we can look at those from the previous season that were underrepresented, and probably we don't have any training in our immune systems for them. Those are the most likely to be infectious. So then we can prepare effective vaccines against those very specific codon changes. And it's always a hit and miss thing, but when your book is published, at least, it was looking at 9 of the last recent flu seasons. They'd done a pretty good job of predicting that accurately. So those are relatively minor changes that do affect the ability of the flu to infect an individual and our ability to develop vaccines. Sometimes, though, viral strains show more radical genetic alterations. This occurs when we get two radically different viral strains inside of a single individual where we get recombination of major RNA components. So for example, if we're an individual working in the swine industry and you get the flu, the human flu, but at the same time you're still working and you're working with a pig that also has a swine influenza, you could get infected by both, and if the same cells get infected with that, you can create a new virus that has viral components of each of those. So this is where we get a lot of these really bad viruses, people working in the swine industry or people in Asia working with large duck herds. This is where we can get some major flu outbreaks. And the 1968 pandemic was associated with one of these situations where we got components, an introduction of H3 proteins from an avian strain into human lineages. So if we look here, the different colored branches indicate viruses that typically infect horses in kind of the greenish yellow. The black branches are those viral lineages that tend to affect birds. But occasionally, again, you can see within a bird clade, a bird virus clade, we see popping up swine. So these pigs would be getting viruses that have a large component of avian viral characteristics. And so it's more likely to evade their immune system, and this is when you're going to get really sick. Same thing happened with humans. And so in 1968, this influenza that caused a pandemic was derived from a virus that was primarily avian, but it was incorporated within humans with addition of human strain components And again, that's when you get really sick. That's when the influenza spreads really quickly because our immune systems just don't recognize it as quickly. And you can't predict those kinds of major alterations, and so typically you don't have a vaccine ready for those types of rearrangements. Let's now move on to talking about the coevolution that bacteria have with us when we're trying to combat them with drugs, which we call antibiotics. So when we take antibiotics, we are trying to kill bacteria. Well, the bacteria are trying to survive, and so that leads to this coevolutionary arms race. And one of the things that you can do to actually make it easier for the bacteria to evolve resistance to the drugs is to not take your medications effectively. So what oftentimes happens is you get a bacterial infection and you get a course of antibiotics to take, and you're supposed to take it, say, for a week. Well, typically what happens is after the first couple of days, the medicine's working and you've killed off maybe 90% of the bacterial population. And so you feel good. Some people then stop taking the medication, but realize you haven't killed off the bacteria completely. And think about it, the bacteria that are still there are the ones that were the most resistant to the drugs. So if you stop taking the drugs, you have a relapse of that infection, and you're getting more of the resistant individuals. And if some of those in their continued reproduction get mutations that make them even more resistant, then you're going to have problems in the future with that bacterial lineage. And sure enough, if you look at bacteria that show resistance to antibiotics, first-time infections, it's relatively low. A relapse of the same bacteria tend to have much higher antibiotic resistance. And that is because you have challenged them, but you've allowed them to succeed, and you've selected for those bacterial lineages that show some antibiotic resistance. What you should have done is take the whole course of antibiotics, and there's a better shot at that point that you're going to kill off all of them. That's why it's very important to go ahead and take the complete course of your antibiotics. Luckily, in some cases, when the drug is removed from use for a number of generations, the bacterial populations will lose that resistance. And this is a graph showing that as being the case in Iceland. So that here was the peak of resistance, and after it was used, after the drug was not used for a while, it did show some declines. But that's not always the case. Streptomycin resistance doesn't appear to decline. It really just depends if this adaptation from the construct of when it's being faced with the antibiotics, it's clearly adapted then. But does it cost the bacteria to maintain that resistance capability when the drug isn't there? That's going to determine whether the resistance is going to decline or not. If it's not costly, they may just go ahead and keep those alleles associated with resistance. And that's going to be a long-term problem for us. So we need to do everything we can to stop this antibiotic resistance from evolving in these bacterial lineages. So how do you do that? Well, here's a paper that came out in 98 to kind of talk about some of these issues. And the first thing is just practice good hygiene. Wash fruits, vegetables, don't eat raw eggs, undercooked meats, and then wash your hands frequently. But try to minimize the use of any kinds of soaps or cleaners that actually have antibiotics in them. I think this is relatively rare. Just use regular soaps. And then, as I mentioned, if you do take antibiotics for an infection that you have, follow the instructions carefully and finish that course of treatment. And this also means don't use these antibiotics when you don't need them. Viral infections, don't take antibiotics. Antibiotics are due to bacterial infections, not viral infections. Now, you may get a viral infection that reduces your immune system's functioning to the point that you develop a secondary bacterial infection, and then yes, antibiotics might be needed. But if you get a cold or influenza, antibiotics aren't going to do anything. If you're in the medical profession, make sure you wash your hands between patients. Show really good hygiene. And we're seeing that this is really important also with COVID-19. And sometimes patients, if they're feeling bad and you tell them that you've got a virus and there's nothing they can really do for you, they get angry and they want something. And so some doctors will go ahead and prescribe unneeded antibiotics just to kind of get them to shut up and get off their backs. But you got to stand up to these insistent patients and explain to them it's not going to do any good and it's just going to be a waste of their money. If you do have to prescribe an antibiotic for a bacterial infection, if you know what that bacteria is, you can prescribe very specific antibiotics that just target that and avoid the use of broad spectrum antibiotics. And lastly, worst case scenario, if you are treating a patient that's not showing a good response to the use of antibiotics and showing some antibiotic resistance, you have to isolate those individuals. They need to be quarantined so that those bacteria won't spread throughout the hospital or the community. So let's talk a little bit more about virulence. I talked a little bit about this in the last video. Why is it that pathogens cause variable degrees of harm to their host? So if you get the cold virus, makes you a little sick, but it's not end of the world. Influenza, COVID-19 can kill you, but even among individuals it can vary. So why is there variable virulence? And remember, virulence is a measure of how fast some infectious particle, either a virus or a bacterium, is growing. And the faster they reproduce, typically, the worse it is for the health of the host. So we're going to talk about a variety of hypotheses that address this question. The first is called the coincidental evolution hypothesis. And the idea here is that the pathogen just doesn't have a long coevolutionary history with the host that it finds itself. It may have evolved adaptations for its typical ecology, and it just so happens to cause harm when it's in you, a host that's not normal. So, for example, the soil bacterium that causes tetanus, it has lots of adaptations for living in soil, and it produces toxins in that context. Those are adaptive in that situation. But if it infects a human, it's still doing its normal thing, and it produces toxins that are lethal to humans. Of course, that's not good for the bacterium. It's obviously not good for the human, but it's because that's not a normal co-evolved relationship. It's just a coincidental introduction of that bacterium into a human occasionally. It's not their normal host. Another hypothesis is called the short-sighted evolution hypothesis. And the idea here is that you have different pathogens, different lineages that are reproducing within the same host, and they're competing with each other. Remember, fitness is a competitive venture, and the virus or the bacterium that is growing the fastest is driving the bus on the evolution of those lineages within that single host. So they have better within host fitness, so it's short-term gains. But if it ratchets it up to the point that it leads to the death of the host before the pathogen can really be transferred to another host, well, that's not adaptive long-term. So there's short-term adaptive, but long-term that isn't a virulence that's adaptive. So we would see in those situations a coevolutionary balance such that it reproduces as much as possible to outcompete slower reproducing pathogens, but it's going to reach a peak where it doesn't want to overshoot that so that it makes the host so sick that it reduces the chance of transmission to additional hosts. Lastly, we'll talk about the trade-off hypothesis. And the idea here is that pathogens that reproduce quickly, they're really high concentration of the bloodstream or the tissue of the host, and so they're more likely to transmit themselves when they make contact with a new host because of that greater concentration. But again, that typically is going to be associated with making their host sicker. So they may kill their current host more quickly. So in many regards, this is very similar to the previous hypothesis, the short-sighted evolution hypothesis. But it's putting the proximate mechanism of why it may limit transmission to the next host. But is that really going to be the case for all viruses or bacteria? Well, we do see support for this idea. That there may be trade-off in some species, but not in others. So if you look at vector-borne infections versus those that require direct transmission of pathogens, we see that vector-borne pathogens show comparable frequencies when they're relatively low virulence and when they're relatively high virulence. Really not much of a difference. It's kind of evenly spread. So high virulence and high mortality is not necessarily a bad thing in these vector-borne pathogens. So think about malaria that is transmitted by mosquitoes. Yes, it can reproduce really quickly, so that it increases the chance that every time a mosquito bites an infected individual, it's going to pick that plasmodium up and transfer it to a new host. In that situation, probably the more virulent you are, the better. And even if it does make your host really sick where they can't move, that may be even better, right? They're not going to be able to swat at mosquitoes. They're going to be in such a feverish state that that's actually beneficial. So certainly there is no trade-off in this situation between keeping your host healthy and keeping just the right amount of pathogens in the bloodstream. However, if you look at directly transmitted pathogens, you see a much higher frequency of those showing very low virulence. Again, here they're having to kind of balance the health of their host for longer so that they can go out and be social and have the potential to meet other potential hosts directly. Same kind of pattern is seen with directly transmitted intestinal pathogens versus waterborne ones. Waterborne ones don't have this trade-off issue. They're like vector-borne. As long as they can get in the water supply and the water supply distributes them, that doesn't matter. So especially if you're talking about things that affect the digestive tract that can cause dysentery and diarrhea, in those situations, the higher the concentration, the better, because you're going to make those individuals go to the bathroom, get into the water supply, and contaminate it so that it increases the chance of finding new hosts. So high fraction of waterborne illnesses show lots of death, and that's because of that water pathway. Next topic is looking about the evolution of human health via diet. If we look at our diet prior to the advent of agriculture about 10,000 years ago, we see that that diet was quite different from the one that we have today. So hunter-gatherer societies, even today, have about 65% lean game, wildfowl, eggs, shellfish, and fish, with the remaining part of their diet associated with fruits, vegetables, nuts, and honey. Relatively healthy diets. Now, we still have those two categories, but much reduced in frequency to about half. The remaining half of our diet is consuming saturated fats, alcohol, artificial sweeteners and sugars, higher fat content, cereal, processed cereals, milk products, which were non-existent in these societies or in very rare occurrence. So there is evidence that hunter-gatherers will consume alcohol, but it's not nearly as available as it is in modern societies, for example. So is this why we see issues with disease in modern human populations? Sure enough, our modern diet is linked to heart disease, stroke, and cancer. These illnesses are relatively rare in hunter-gatherer societies, so there could be some link here. But realize there are other differences between our societies and these others, hunter-gatherer societies. Certainly they get more exercise than we do. And so that in itself may play as much of a role as the diet itself. One of the things that has changed a lot through human history is milk drinking in different societies. In some populations, it has led to the extended production of lactase so that we can digest this milk. Most mammals, they stop producing lactase in individuals after they wean. They're not going to need that ability to break down milk sugars, and so they stop producing the enzyme lactase. However, human populations with a history of raising dairy cows have evolved extended lactase production. So when it's adaptive, we have seen mutations that have led to this extended lactase production evolving in populations that are in the appropriate context if they're still drinking milk. If they're not drinking milk, you're not going to see selection for that proximate mechanism. What is the mutation rate in humans? We talked a little bit about this when we talked about mutations in general. But if you look at how these accumulate in humans, this study by Lynch et al. In 1999 said that the total mutation rate was about 100 to 300 mutations per individual per generation. But again, many of those are going to be silent substitutions and not really have much of an effect. They estimated that deleterious mutation rate was about 2 to 7 per individual per generation. And that slightly deleterious mutations can actually increase in human populations by about 4% per generation, assuming that there's no selection to get rid of these slightly deleterious mutations. Well, why would that be? Remember, natural selection is pretty powerful and it can get rid of even slightly deleterious mutations over time. So why would you assume no selection? Well, the traits that they were talking about in this paper, it does make sense. So visual acuity, tooth alignment, childbirth problems, infertility. These types of mutations can accumulate into a population of humans because we can remove selection. We have modern medicine. We have the ability to wear glasses or contacts. So I'm a perfect example of this. I can't see anything without my glasses or contacts. But it doesn't affect me at all because I can just use modern technology to get rid of that selection pressure. Tooth alignment, the same thing. I had horrible teeth, got braces, no big deal. Childbirth problems. 100 years ago, women not infrequently died during childbirth because of medical issues. Now we can have C-sections. And that's a good thing. But that does mean that there is an accumulation of slightly deleterious mutations that are going to more likely have these things pop up more commonly in future generations because we're not selecting them out. Same thing for infertility. We have all of these infertility treatments. Let's just look at one of these, visual acuity. Myopia is seen in about 25% of the people in some populations. And so this is nearsightedness. So you can ask the question, well, maybe nearsightedness just isn't heritable. If that's the case, you could see it accumulating in populations because it's maybe just a plastic trait that's all environmentally determined. Well, twin studies show that there is at least some heritability. If you look at monozygotic twins, you would expect both of them to be myopic more often than one of them being myopic. And that's exactly what you see. But there's still, it's 2/3 this way, 18 versus 36. So there's still quite a few of them where they're identical twins, but only one of them is nearsighted. So that tells you that there could be some environmental component. And sure enough, in dizygotic twins, just typical brother and sisters of the same age, oftentimes you see that only one of them is myopic. So there may be some heritable basis to this, but there's probably some environmental aspects to blame too. For example, if you look at individuals that have gone through some cultural shifts, you see that we see a change in the percentage of individuals that are myopic. So in northern Canada, there was an Inuit study that looked at two different age classes of individuals. Older individuals relatively rarely were myopic. Those that were raised during the time frame in which modern schooling was available, much higher fraction of those were myopic. So that's due to just reading more. Breast cancer is an illness that has a genetic basis in some cases. There's strong evidence to support heritability of this so that certain female lineages and even males can get breast cancer. A good friend of mine had breast cancer. So we know that there is a heritable basis for this, but there could also be environmental influences as well. Turns out there is a mouse that carries a virus that if it infects human tissue, it can also increase the potential of those hosts of developing breast cancer. It's called the mouse mammary tumor virus, and if humans are exposed to the MMTV, it increases their chance significantly of getting cancerous tissue. If you look at normal tissue, you tend to not see the MMTV tissue. Now, there are two species of mice that live in Europe, and one of these tends to be associated with a higher rate of this MMTV virus. The mus domesticus, which is seen more in Western Europe, is the main house mouse there, and that's where you tend to see these higher incidence of breast cancer. In Eastern Europe, where you tend to see mus musculus, you tend to see lower rates of breast cancer. Now, it's my understanding that both of these species have been introduced into North America, so your typical house mouse is in the genus Mus, but I believe that most of the ones that we have around here at least are Mus musculus. There's another environmental factor that may contribute to the likelihood of developing breast cancer. Continuous menstrual cycling is associated with increased levels of breast cancer. This is because during the post-ovulatory phase, there's stimulation of cell division in milk ducts. So the more cycles you have, the more cell division you have, and that's when you are more likely to get mutations and more likely to have some of these mutations be the cause of cancerous tissue. If you look at the Dogon women of Mali, they have about 1/3 less number of cycles as women in developed countries. And you can see that during this time frame, they tend to not have much menstruation. And the reason for that is if you look over here, many of them are breastfeeding. That's what this category here means, or pregnant. So only about 1/4 of this proportion of women at any one time is cycling. The others are pregnant or nursing, or they're old enough that they're post-menopausal. But this greatly reduces the number of menstrual cycles they have in their lifetime. And if you look at their breast cancer rate, they're about 1/12 of those in modern societies that are showing continuous cycling. So now let's look at a physiological response you have to many infections. You get fever. If you look at the function of elevated temperature in ectotherms, it is used to fight off bacterial infection. We call this behavioral fever because these are ectotherms. They have to go to hot areas to artificially raise their temperature higher than normal. So this is seen in a lot of herps, fish, invertebrates when they get sick. They go to these hot areas as a means of fighting these infections? Well, we can get a fever because we're endotherms by ratcheting up our metabolism. So this begs the question, is fever an adaptive response to an infection? And if so, if we take medicines to reduce fever, is it actually making things worse? Well, there have been studies to address this, and they're inconclusive. And so the main thing I want to get across to you here is, while this is an interesting hypothesis, fever is a tough thing to control, and you can get fever-induced tissue damage, and that may outweigh any potential benefits of letting your fever get high. Particularly if you get a really high fever, it can affect the brain in ways you don't want it to. And as I mentioned, in endotherms, high temperature is also linked to higher metabolism. So higher metabolism, higher immune response functions, and fever could each play a role, and it's hard to disentangle which of these is playing the biggest role in combating infections. In one study, it looked like increased metabolism, but not necessarily fever, was linked to an increased immune response and function in rats. So maybe just jacking up your metabolism is the way to go. Some studies have indicated that anti-fever medications can harm the functioning of your immune system independent of its effect on fever. So here we look at a study of the common cold, and people that were taking aspirin, acetaminophen, and ibuprofen had greater problems with obstructions in their nose. So they're more snuffly. Individuals with placebos actually did the best. And if you look at the production of antibodies, again, antibodies are going to be helping you to fight off these infections. Placebos did better. They produced more antibodies compared to ibuprofen, aspirin, and acetaminophen. And if you look at the mean changes in monocyte counts from the baseline levels, we see monocytes being produced at a much higher rate in everything except the placebos. And lastly, I just want to talk a little bit about parental care. We talked about parental care as being a costly trait, and that most organisms don't show parental care to their offspring as one of their life history goals. But there is some potential conflicts of interest between parents and offspring at times, when the parents are trying to save resources for future reproduction and the offspring may be wanting more of the attention. So even in the best of circumstances, parental care is costly, and parents have to dole out the parental care in such a way that it maximizes their lifetime reproductive success, even if it may be costly to individual offspring. Well, if you think about parental care in that regard, you wouldn't be surprised to show that the data show that a variety of animals treat their genetic offspring and step offspring quite differently. And that's the same case in human populations as well. I mean, this is the reason why we have so many fairy tales and folk tales associated with the evil step parent. There are data to suggest that step parents don't treat their step offspring as well as their genetic offspring. Here's a study from Trinidad in which they looked at how fathers interact with genetic offspring and their step offspring, and they interact more with their genetic offspring than their step offspring. Now, that could be an indication of care. The more time you spend with your offspring, the more care that they're receiving. But if you just look at the percentage of agonistic interactions with genetic offspring and step offspring, the genetic offspring don't receive nearly as much agonistic behavior. Agonistic behavior are aggressive, mean behaviors, confrontational behaviors. The step offspring, they don't get much attention, but when they do get attention, it's not good attention. In Dominica, there is a clear pattern that step offspring have higher cortisol levels. Cortisol levels are, again, indicative of high stress levels. These are stress hormones. And having a higher cortisol level is greatly linked to number of days ill. And so you're going to miss school. Your general health is going to suffer. And again, this is being linked to them not receiving proper parental care or more stressful interaction with step parents. Now, this study did an interesting thing, and you may be thinking, well, that may be just linked to households that are relatively poor and not educated. Well, in Dominica, they broke it down by social class, and higher class versus middle class and lower class individuals, there was no difference. You still see the same pattern, that the step offspring are treated worse and they are ill more because of having high cortisol levels. And this can extend even into the next generation. If you look at the eventual reproductive success of stepchildren, they have lower reproductive success. So if a female that eventually reproduces didn't have a step parent, they have higher reproductive success than those individuals that did have a co-resident step parent. Same thing, even a greater, more drastic change in males. Males that had a step parent have lower reproductive success when they mature. And lastly, stepchildren sadly suffer much higher murder rates. So if you look at the murder rate of young children, particularly the youngest children, they suffer the highest rates of murder at the hands of a step parent. You do see this very rarely in natural parents, but it's nothing compared to what you see in the step parent situation. Now, patterns like this are to simply show you what past evolutionary traits may have been in human populations. Some people have taken this as an excuse, saying, well, this is what my genes have evolved to do. That is not what I'm saying. This is not ever going to be used successfully in a court case or to use to excuse bad behavior, because we do have the ability to think through our behaviors and make appropriate choices despite our potential evolutionary past. And actually understanding the potential for these behaviors can help social workers spot potential troubles and step in at the right time. So there's one more video in this series, and in the last video, obviously any evolution course wouldn't be complete if it didn't cover human evolution. So that'll be our next topic.